Hi everyone, my name is Jeongbin, a PhD candidate in the Department of Computational Medicine and Bioinformatics. I, as a member of Single Cell and Spatial Analysis Forum Task Force Trainee, will present a seminar named Spatial Transcriptomics and Drug Development. So before I jump in, I'd like to mention the market characteristics of pharmaceutical market. As of 2024, the pharmaceutical market has a size of $1.6 trillion, while the global semiconductor market has about 800 to 900 billion dollars in size. So the size differences are about 2 times. So we can say pharmaceutical market is the largest market in the world. And as of 2023, Novo Nordisk, which developed a obesity drug named Wegovy, has a market cap of $412 billion, which is first rated in Europe, and which has increased 300% from 2022. Note that the global domestic production, or GDP of the Denmark, was $406 billion. So the market, the stock price market of the Novo Nordisk, which is in Denmark, is higher than the global domestic production of Denmark, which is very interesting to me. Also, at the same time, Novo Nordisk had operating profit of $14 billion, which had increased 30% from 2022. So we can say even a single drug can sustain a nation economy. Very recently, I had a very interesting news. So Maunjaro, which is developed by Eli Lilly as a obesity drug, surpassed the sales of Keytruda, which is immune checkpoint inhibitor for cancer therapy, and became a #1 selling drug. So actually, the drug market is very dynamic, and also the market size is very enormous. So please note that the overweight and obesity population out of total population is expected to increase, and also the obesity drug total market can increase, as can be seen here in the following years. So the economic demand from the customers regarding the drug and pharmaceutical is getting increased as time goes. Then how do we cope with that increasing economic demand from the customers in the world? I do believe that data drives the growth in biopharma industry, and they can cope with the increasing demand of the customers. Please note that the size of the genomics data is the largest one when you consider 4 different data modality. Here, astronomy has 1 exabyte per year in generation, and also the Twitter will be generated about 1 to 17 petabyte per year, YouTube 1 to 2 exabyte per year, and also in the case of the genomics, it will be generated to 40 exabyte per year. Can you imagine what exabyte really means? Exabyte refers to the 10 to the power of 18 byte, which is 1 million terabyte and also 1,000 petabyte. Please note that 1 terabyte corresponds to 1 hard disk, while 1 petabyte corresponds to 1 big server. So maybe every year we may need to have 1,000 big servers to store that amount of genomics data yearly. However, even if there are a lot of data that can cope with the increasing economic demand from the customer, the problem is the unstructurization of the data. So there are a lot of data set in the bioinformatics and biological sciences, including molecular data, cellular data, histological data, and even the clinical data from the medical doctors as a form of nature descriptions. And I think various types of data are being integrated around the spatial omics. This is the figure plot I obtained from the spatial glue paper. And here we can think of transcriptome data, epigenome data, proteome data, or even morphological data with the natural descriptions. And they can be incorporated into one single spatial coordinate with the help of the data integration algorithms. So I think spatial information is very abundant and very, for example, dynamic, so that a lot of data modality can be incorporated into one coordinate or one single modality. And I do believe spatial omics can provide the potential. And due to that advantage of the spatial omics, including the structurization of data, and so visually attractive, and also intuitive, and also the potential to provide a serendipitous discoveries, I think spatial omics are gaining popularity for drug development or even biological sciences overall. So spatial transcriptomics was chosen as the method of the year in 2020. And also the extended version, the spatial temporal analysis for embryogenesis, was chosen as the method of the year in 2023. And also the spatial proteomics is chosen as the method of the year in 2024. And this year, February 6th, Chan Zuckerberg Initiative, which is the greatest funder for the biological science and bioinformatics, launches billion cell project with 10x Genomics and Ultima Genomics to advance AI in biology. And actually, the objective of this project is to bring about the single cell and spatial technology to understand the complexity of biological science, potentially for drug development. The CEO of the third is Saxonov. I also personally met him in the previous academic conference. Once said that single cell and spatial technologies are the most significant transition. And he also believes that single cell and spatial technologies can contribute to the drug development process, which is aligned with my message here. So actually very interesting trend has been observed. So Illumina sequencing is, as you know, very popular technology for short-lived sequencing. And Illumina decided to announce new data modality, spatial technology, spatial transcriptomics technology. As it has its own foundational technology, it can provide very large region of interest, which is 15 by 50 millimeter square, which is much larger than the most up-to-date, and the largest ROI of Visium HD extended version, having 11 by 11 millimeter square. And also Illumina has announced that it will develop its own bioinformatics technology to delve into drug development. So maybe, as you can see, 10x Genomics products are mainly based on the Illumina sequencing as well. So there may be some conflict, isn't it? So October 21st, 10x decides to sue library Illumina over gene sequencing patents. And I do believe that the increasing economic demand may open new market opportunities, and maybe the next winner should take that trend. So Illumina, 10x, or other companies are now interested in that type of trends to be the potential winner in the future. And also, even the virtual cell trends align with spatialomics and drugs. This is the article I observed from the Time magazine, which is published in October 14th. And it says, AI companies are racing to build a virtual human cell. And there's a history for that emergence of concept. FDA, the Food and Drug Administration in US, announced that this year, it will discourage the use of animal experiments and encourage for example, alternative technologies, including organ-on-a-chip systems, computational modeling, and advanced in vitro assays. I think virtual cells are somewhat summarizing these keywords, and there may be some potential reasons underlying it. The first one is ethical issue of animal experiment. The second one is economic cost of animal experiment, so that we can easily decrease the cost of drug development when we have alternative technologies. And also the third reason is AI. AI is already measured, so that FDA can think of the use of AI in the drug development process as well. This is the example figure obtained from Noetic, which is also dedicated to virtual cells. And here we can see anti-PD-1 responders and non-responders. Let me give you a context about it. So immune checkpoint inhibitor means the drugs that can target the immune checkpoint blockade of cancer cells that are evading from immune cells. However, for example, if there are terminal cancer patients and the immune checkpoint inhibitors are that effective, then the patient can be cured. However, the percentage of the treatment responsive patients is about 15 to 30%. So the thing is, we need to identify which patients are responsive to this type of very effective drugs. So the responder prediction is very important in this context, and maybe virtual cell models can simulate which type of patients are responsive to this type of drug or not. And I think the virtual cell technologies are now combining with spatial omics technology and also other drug modality or other drug-related data as well. I will introduce such a data in the following slides. And also, as you know, some AI companies like Anthropic and also OpenAI are interested in this type of pharmaceutical, biotechnology, or life sciences. And actually, they are all in the same page. And I'd like to introduce examples and my research experience about it. And so I think you may or you may not part of the HRD, or homologous recombination deficiency. HRD was first approved by US FDA for niraparib and its application in ovarian cancer on 2019 October, and its approval expanded to other companion diagnostics as well. So currently, clinicians obtain the blood samples or liquid biopsy samples from the patients and then perform the genomic analysis and then compute the copy number variation, compute the HRD score, and then make a decision to prescribe niraparib or other drugs to a certain patients. The problem of this technology is the price. At the moment, the price is for $4,500 per kit. It's not cheaper. So maybe alternative technologies may be encouraged to provide democratized health, or as you know, yeah, there are a lot of precision medicine or personalized medicine trends. And actually, however, we can have a lot of copy number variation estimates and algorithms from diverse modality, including whole genome sequencing, single-cell DNA sequencing, single-cell RNA sequencing, even spatial transcriptomics. Here, CalicoST was developed by supervisor Songma, and also some other single-cell RNA-seq-based copy number variation algorithms can be applied to spatial transcriptomics as well. I've seen that type of trends multiple times. So the idea is here. We have spatial transcriptomics data. We can predict the copy number variation, and we can compute the HRD scores by summing up the three different scores: LOH, loss of heterozygosity, LSI, large scale state transition, and TAI, telomeric allelic imbalance. By summing up such three scores, we can result in the HRD score, as can be seen here. And so we have a pair of data, histological images and HRD score. Then we can make a digital pathology model that can predict the HRD scores from the histological images. So if a new histological images are given, then maybe we can predict the HRD scores easily. Then maybe we can make a decision to prescribe a certain FDA-approved drug or not for that patient. Please note that this technology is very cheaper compared to the liquid biopsy sample. Needle biopsy is there. So the clinician has a needle and he puts the needle into the patient and then pull it out. And then we can obtain the single, histological tissue. And then we can perform the H&E staining when we can scan it and we can obtain the histological images easily. And if models are there, digital pathology models are there, we can compute the HRD scores in this way. So maybe it should be cheaper. However, I want to mention one thing: the digital pathology is very hard, even if it has a higher economic potential, because the digital pathology model should be resolution agnostic, staining protocol agnostic, institution agnostic as well. So it's not easy. So even if it has a lot of economic potential, and so many companies, including Okin or other Microsoft or Google, are interested in this type of technologies, there is no single success at this moment. But maybe in the future, who knows? And I'd like to introduce another example. So from now, in this page, I'd like to mention the bispecific antibody drug conjugates. Antibody can be attached to the drugs, which is called antibody drug conjugates. And it has two different mechanisms. The first one is end manner and end way, and the other one is OR gate strategy. So for example, if there are two different targets in a target cell or tissue, then in spatially closed in a closed space, then maybe the ADC can target such a cell. This is what AND gate strategy refers to. And also, if either of two targets are expressed in a single cell or cancer cells, then ADC may act at the cells and kill the cancer cell or something like that. In such a case, it is called OR gate strategy. And here there is an example. So we can think of multiple cancer cell lines, and the sensitive cancer cell lines to a certain drug may have IGF, insulin growth factor 1 receptor and insulin receptor heterodimers. But in this case, for example, IGF-1R and IR are co-expressed in such a cell, then maybe the bispecific antibodies acting as AND gate manner can kill the sensitive cell lines. So this is the one example of the potential of bispecific antibodies or conjugates. Actually, this has different literature, but I can suggest, bispecific antibodies can act at this context. And this is another example I was involved in. So we can find the necrotosis related hepatocytes and also macrophages, and compared to the normal liver in the acetoaminophene or APAP induced acute liver injury tissue, we can find a higher spatial overlap between necrotosis related hepatocytes and macrophages. So if a certain drug can act at macrophages, then maybe we can also affect the interacting or co-overlapping necroptosis-related hepatocytes. So the very initiative or the motivation of this research is to develop such a drug. And before that, they are trying to find such a spatial overlap pattern in the disease tissue. And I was involved in, and I made this plot for them. And also, this is another example I was involved in. It has, for example, we can think of this title is pharmacokinetics. So spatial omics can identify the specific biological factors that are related to the pharmacokinetics, which is also called drug distribution. And here we can think of multiple albumin platforms, which has different glycation. So here mannose, galactose, and glucose labeled albumin can be synthesized. There is a mechanism of this type of thing. So we can prepare the glycation related azide here. And also albumin has its own, albumin has its own alkyne. So the azide and alkyne can form a cycloaddition reaction in this way. So actually in this way, there is heat is needed in this way, but in the recent technologies, copper ion or other catalysts are involved so that we can make such a novel drug modality in the in vivo friendly environment. And as we have different albumin platforms with different glycation, we can find the spatial distribution in the spatial transcriptomics, and we can compare which factors are related to the distribution of the albumin platforms. And here is the example. So mannose albumin, glucose albumin, and galactose albumin are here. And as you can see, mannose albumin has a preferential distribution in the lower part, which corresponds to anti-inflammatory macrophages. It is as expected because mannose, M2 macrophage or anti-inflammatory macrophage has a certain receptor that can take the mannose albumin primordially. And while the glucose and albumin and galactose albumin platforms are primordially located at the right side, which corresponds to cancer cells or inflammatory macrophages. In another tissue, it is observed reproducibly. For example, here mannose albumin has preferential distribution in this way, and here it corresponds to anti-inflammatory macrophages as well. However, in the case of glucose albumin and galactose albumin, their distribution are somewhat remarkable in the lower part, which correspond to primordially cancer cells here. So please note that cancer cells have higher glycolysis rate, which is called Warburg effect, and also metabolism is very effective. So cancer cells need a lot of glucose from other surrounding tissues or bloodstream. So that's the reason why even a glucose attached albumin can also be recruited in the lower part. Please note that albumin is very big molecule. However, the single glucose is attached to the albumin can change the distribution of such a drug in this way that effectively. And this is another example which is published in Small Methods, and actually I was very proud of it because it is officially my first author paper. And we have histological images, HN images, and drug distribution and the corresponding spatial transcriptomics data. And as you can see, here, please note that instead of the drug including the payloads, it is just the vacant liposomes or lipid nanoparticles labeled with DID lipophilic dye. And so there is no such a drug-related cell killing effect or not, or something like that. The purpose of this research is to identify the biological factors that are governing the distribution of liposomes or lipid nanoparticle. And here, as you can see, the peripheral uptake pattern of the lipid nanoparticles is observed, which corresponds to the stromal part of the cancer. And also, when you define the distance from periphery to the inner core of the tumor, the accumulation of the fluorescent lipid nanoparticle is somewhat unexpected or unintuitive. There is a very complicated pattern. I just searched the endocellular or blood vessel markers, including PKM1 and CD34, and I simulated such blood flow-related passive uptake of lipid nanoparticles within the tissue with the peak simulation. However, that patterns only correspond to the peripheral or lower part of the tissue, not this inner part of the uptake pattern. And in the later slides, I identified that the cancer-specific metabolisms, including glycolysis, apoptosis, or hypoxia pattern are related to this heterogeneous uptake pattern of the lipid nanoparticles. And I do believe that cancer cells are having motivation to recruit a lot of molecules surrounding the tissue because it is very congestive area. So many cancer cells are within the smaller compartments so that a lot of cells are dying, but a lot of cancer cells are also proliferating. So maybe very active metabolism is involved, and the uptake or accumulation of the lipid nanoparticles may be related to that. That is the conclusion of this paper. And for example, here, I obtained the drug distribution image in the neighboring slice. So as you can see, the boundary of the drug distribution image is somewhat different from the histological images. So maybe to perform the drug distribution related, to identify such a genes involved in drug distribution, maybe we need to perform some alignment of two different modality. And here I align the spatial transcriptomics with the drug distribution images. And the idea is there. So spatial transcriptomics, which can be thought of as a kind of tabular data, can be Gaussian blurred so that we can make a certain two-dimensional image. And there are a lot of 2D image alignment algorithms. And I was involved in the development of open source Python DIPI package-based image alignment algorithms to perform, to identify the genes that govern the distribution of the drugs. And also, I heard a lot of comments about the temporal information about this research because, for example, here we just only have one single time point drug distribution image, but we may be interested in the drug distribution in the temporal manner. So the following research is to address the temporal information and maybe the potential to be combined with the spatial omics data. And here we developed, DID labeled lipid nanoparticles and treated them to the 41 cancer cell line. Please note that the tissue is also 41 cancer cell tumor model. And here, as you can see, at the first time, the first 20 minutes after the treatment of the drug, many drugs are primarily located at the cell membrane of the cancer cell. However, as time goes, the lipid nanoparticles are migrating into the nucleus. So the spatial distance between the nucleus and the lipid nanoparticle are getting closer as time goes. And maybe during that time, the endosomal pattern has been different. For example, at the initial phase, early endosome is involved, and at the later phase, late endosome or even lysosome is involved in the uptake pattern of this. And also, I loaded iron nanoparticles in the lipid nanoparticles and treated them to the cancer cell lines. And as you can see, at the 40 minutes from the first treatment of the drug, there are a lot of lipid nanoparticles containing iron nanoparticles. However, as time goes, the abundance is getting decreased. And also there is some exocytosis pattern. So actually the temporal dynamics of the drugs is very complicated, and maybe it's desirable to combine them with the spatial omics data. And here is another data. This experiment is very similar to small molecule fish, and I prepared two different drugs. DID labeled fluorescein lipid nanoparticles and Cy5 hybridized RNA encapsulated in lipid nanoparticle. And here, as you can see, the lipid nanoparticle abundance is somewhat constant across the time. However, as time goes, the internal cargo, the Cy5 RNA encapsulated in LNP, is starting to be released to the cytosol of the cell. Please note that when Cy5 RNA is encapsulated in lipid nanoparticle, there should be some fluorescent quenching effect, so that at the initial time point, we could not see any fluorescent pattern. But after time goes, we can find the released Cy5 RNA fluorescent pattern. So this is another example of the temporal dynamics, and very complicated. And this release pattern is called endosomal escape. And maybe the objective of this research is to identify the rate or velocity of the endosomal escape. And here is another example. So we are interested in the muscle tissue. And as you can see, there is a very typical structural pattern of the muscle cells, as can be seen here. And at the initial time point of the administration of the iron nanoparticle loaded lipid nanoparticle, the nanoparticles are primarily located at the interspace between two different muscle cells. As time goes, the iron nanoparticles and lipid nanoparticles are migrated inside the muscle cell, and then they disappeared as time goes. And also the IBS in vivo imaging system results show that the lipid nanoparticle accumulation is constant over time, just like this outcome. And also, however, the magnetic resonance imaging is somewhat changing. Actually, this is a little bit slight to identify the difference right now, but when we quantify that signal difference in the magnetic resonance imaging, actually there is a signal difference just due to the iron nanoparticle state changes. However, can you imagine spatial transcriptomics and temporal information? It's not easy because of cost. I've seen at least 4 different studies for obtaining 3-dimensional spatial transcriptomics. I mean, having multiple slices of 2-dimensional spatial transcriptomics, that's one strategy. And the other strategy is to have a G-focus related spatial transcriptomics, which is called thick tissue morphish. There may be some recent technologies for that, but at this moment it's very new. However, to combine the spatial transcriptomics and maybe temporal information of drugs, there is another approach, which is using physics-informed neural network, or PINN. At this page, we can think of multiple differential equations, including ordinary differential equation or partial differential equation, to model the dynamics of the uptake of drugs or distribution of drugs. And by putting that differential equation into the loss function of the model, we can make a certain machine learning prediction that is very well aligned with the physical world of us. So this is an example of GFUNC, and it utilizes a kind of a PINN strategy to model the diffusion pattern of a certain molecule for the heart. And in addition to the kinetics. We can also simulate the pharmacodynamics, which is related to the drug mechanism of action. So very recently, there is a very interesting data set called Tahoe 100 million. In this way, we can have a lot of drug perturbation data across different cell lines, different conditions in a systematic way. So it doesn't require our human level labor. It is very automatic, so that the data obtained from this technology can be very accurate. So here, for example, we can find genes, its gene expression changes, drug, sample, and multiple conditions. And then we can make a perturbation effect prediction model from this, a lot of data sets. And now it is evolved into the newest one, Tahoe X1, which is published in October 23rd. And also there is another paradigm for predicting the drug action, which is foundation model approaches. So as you can see, there are a lot of models, including SCGPT, Jeongbin, SCBird, and so on. They are all related to large language models, but they already were aware of the biological context as well. And the most important benchmark includes the perturbation prediction as well. So maybe for example, cell type annotation, they can be done by human labor. However, perturbation prediction is very unintuitive task, and maybe so many people are interested in such a perturbation prediction task by using foundation models. I was also involved in the development of the foundation model, which is Llama. I mean, so I have good understanding of this field as well. So we can, for example, exemplify 8 different models. And most of the models, 6 out of 8 are based on the transformer encoder, and most of them are basically based on the BERT architecture, which is developed by Google. And also 2 out of 8 are based on the transformer decoder. As they are different in architecture design, the pre-training strategy can also be different. For example, if a certain foundation model is based on transformer encoder, the pre-training scheme may rely on masked language modeling, or MLM. While some other models, depending on the transformer decoder architecture, may rely on next word prediction, or NWP. And the fine-tuning strategy can also be different from the pre-training strategy. It can be same, but it can also be different, like reinforcement learning, or cosine similarity-based different learning scheme can also be considered. There may be some pros and cons for its foundation model. For example, 6 out of 8 models depend on the quantitative gene expression values, but that dependence may be susceptible to batch effect, because, for example, we can think of very different gene expression values across different batches, different patients, different conditions. So if a certain foundation model relies on the quantitative gene expression value, then batch effect susceptibility can increase. Cell-to-sentence versus Llama, which rely on just gene lists, top expressing gene lists of a single cell, can avoid that problem. And please note that Jeongbin here has a specialized normalization scheme, which is median normalization and also gene linking textualization. So it has a little bit different from other models as well. So I just represented it as a triangle mark. And also the metadata availability is also important. Half of them don't provide of the availability of the metadata, while the other 4 has provided the functionality to incorporate the metadata. For example, transcriptomer can provide a contextualized gene embedding, so that we can incorporate natural language description, like doctors' natural languages, or conditions, or morphological image information can also be incorporated into this type of model. And also the accuracy and the time complexity can be a kind of benchmarking, the important factor for benchmarking as well. And here, for example, SCGPT has a newest version, which is called SCGPT Spatial, which can provide the application to spatial transcriptomics. And also a cell-to-sentence has its newest version named Cell-to-Sentence Scale, which is developed by Yale University research team, and it has a collaboration with Google, and they just announced a new model they can predict or discover novel potential cancer therapy pathways or strategies with the power of foundation models. Actually, this is aligned with my messages, the spatial omics and also drug development. They are interested in this type of trend. And actually, I do believe the spatial omics raw data can reveal drug mechanism of action. So here, for example, we can have a certain human-derived stem cell or human-derived cell therapeutic agents, and then we can put them into a mouse tail, and then they migrate within the blood flow. And also, we can prepare another mouse models with different disease condition. And as a result, the human cell distribution within the mouse tissue is only observed with the experimental condition as expected. Here I perform customized alignment algorithm I developed by myself. And interestingly, there is a very spatial overlap pattern between mouse endothelial cells or blood vessel cells and human stem cells. And this is as expected. For example, can you imagine a cancer patient have a very higher occurrence of the metastasis in the lung? So the cancer cells can migrate within the blood flow, and then they can be entrapped by the lung vasculature, just like this way. So for example, here, the distribution is very aligned with the endothelial cells. So that can explain why there are a lot of lung metastases happening. And here I developed my own custom alignment because Space Ranger cannot be easily tuned. We cannot easily change the parameters inside it. So I just kind of make my own modification to make flexibility and to increase the statistical power of the detection of human cells inside the mouse tissues. And here, as you can see, Visium fresh frozen data can reveal the microbiome signals. Please note that Visium data, the RNA data, are all polyadenylated. That is principle. However, for example, we can think of the mitochondrial RNA expression within the Visium data. So even if the very purpose of the Visium is to capture the polyadenylated RNA population, there are also another RNA species that is not polyadenylated. So we can detect the mitochondrial RNA or even microbial RNA as well. Here I used 3 different approaches: Space Ranger, custom alignment, and PATH-seq, which is developed by Broad Institute, and also a mapped RNA population, which is not a map to the human, for example, GRCH38 or HG19 or 38, something that type of human reference. And also, we developed 2 different drug modality, LC-lipo and ST-lipo. Here, LC targets the intestine of the mouse, while ST targets the spleen primarily, just by with just different composition of lipid components in the lipid bilayer. And here we we prepared the ST-lipo and we encapsulate TD tomato mRNA, and then we injected that lipid nanoparticle inside the tail of the white mice, and then they circulated and they arrived at spleen tissue. And here, as you can see, compared to the control, in the tissue with the treatment of such a lipid nanoparticle, we can find the primary, the specific distribution of TD tomato mRNA. And this is a quantified RNA spatial feature plot of TD tomato. And please note that we already know the exact sequence of the TD tomato. But the actual RNA population within the real mouse tissue is somewhat different. There's a mismatch of the bases. So I quantified the mismatch values, and as a result, I could find a higher mismatch values at the periphery of the spleen. So we can think of a higher occurrence of RNA degradation pattern at the periphery of the spleen. And this can also be applied to different sequencing technology as well. So I'd like to mention the high-load sequencing. This is a technique for obtaining the transcriptome RNAs adjacent to specific protein. So the step 1, attach a hollow tag domain to a specific target. And step 2 and step 3, it can make its own Laricar species by using the halogen its own reactivity. And then the alkyne handle radical combines with RNA radical and react alkyne RNA with biotinazide to produce biotinylated RNA, and then separate it by using affinity chromatography with stratigraphy. But I'd like to mention that if we follow this scheme, we can skip the step 4, 5, 6, and 7, because if there is a RNA degradation pattern, we can use this type of technology to obtain the transcriptome of RNA adjacent to a specific protein. That's my suggestion, but it's not realized at this moment. And also there is another data. So we can think of human leukocyte antigen has a lot of isoforms, and in the RNA isoform, there are 7 species. In the protein species, there are also another 7 species, but they are not corresponding one-to-one manner. And here I'm focusing on the RNA isoforms of the HLA-G. And here, as you can see, please note that HLA-G 201 to 205 are protein coding isoforms, while HLA-G 206 is not translated to protein. It's also, HLA-G 207 is also not translated to the protein. And please note that here in the malignant cancer cells, there are a lot of protein coding RNAs, while the other cells are not. So for example, if we anyhow synthesize a certain drug that can target the proteins of HLA-G, then we can easily imagine that will primarily target the malignant cancer cells. So take-home messages are as follows. Pharmaceutical market is the largest market, so many research aims it. And unstructured data is a big hurdle, but spatial omics can provide a basis. And spatial omics can advance pharmaceutical research in many ways. And I'd like to mention some perspectives as the last slides. It's not last, but it's very close to the end. So I'm only focusing on the reading perspective of the drug. For example, we should identify the distribution of drug or mechanism of action of drug and so on. They are all reading perspective. And what about writing? What about synthesizing it? Do you believe drug synthesis is that difficult? To be honest, it's very easy. There's very automated way to synthesize drugs. For example, one example is a PASI display, which is also Nobel Prize winning technology. There are a lot of viral population. We can make a lot of mutations by using mutation wisely. And then we can collect the peptides from its viral population. And then we can analyze the affinity, the target affinity or binding affinity of it, of the produced peptide from the biter, and also the cancer target of interest. And then we can find a specific viral population of interest that may be potentially good for drugs or even some other therapeutics. So in this way, we can find the drug generating viral population in a very automatic way. So that is one example. And also this is another example. For example, in our department, there is another professor named Indica, which is also dedicated to automatic lab. And in 2023, it's already published in Nature. It's in the UC Berkeley, which is called A-Lab, automatic lab, so that it can synthesize a lot of drugs in a systematic way. So, for example, if we have our hypothesis, then maybe we can validate it in a systematic way without any human intervention. So, also there's a lot of drug modalities from small molecules, nucleic acids, peptides, enzymes, antibodies, cell therapies, microbiome in the order of complexity. And please note that, for example, small molecules are now losing the popularity because we have already tried a lot of things for the small molecules. And also, it's very hard to be automated. However, in the case of the antibodies, rather complicated drugs, they can be, for example, automated. For example, can you imagine a drug, a lipid nanoparticles encapsulating RNAs? If we change the nucleotide sequence inside the lipid nanoparticle drugs, then maybe we can make very different drugs. So in this way, the automation of the drug is very important trend in this scene. For example, now Novartis is dedicated to the radioligand therapeutics, and also AstraZeneca is very dedicated to antibody drug conjugates and so on. They are not interested in small molecules anymore. And also I'd like to mention the antibody perspective. So for example, we can model the structural biology stuff. So there are a lot of open source structural biology machine learning model, including AlphaFold 2 and also BORCH 2. And recently, BORCH 2, which is developed by MIT team, has announced BORCH gene to make the recommendation of a certain drugs from a target by using the structural biology AI tool. So in this way, maybe in the future, the automation of the drug synthesis may be encouraged and also maybe futuristic. I think maybe in 20 years, in 10 years, we can see very popular automation of the drug synthesis and so on. So reading, I already mentioned that. Writing, I already mentioned that. So I think to connect them, maybe thinking is there, right? So computational model, AI model, that's our mission, isn't it? So that's my perspective. And we are single-cell spatial analysis from task force training, and I welcome everyone here. And also we are recruiting many other members, new members. Yeah, thank you.